Okay, hello everyone. A very good afternoon to all of you. And today I would like to present my topic on the how do we actually combat the progression of hepatic steatosis, or we call as the fatty liver disease, into chronic diseases such as type 2 diabetes mellitus, or even CVD, or even obesity. And how do we actually combat this fatty liver disease by using phosphocholine complex? So basically my topic today is more on the effectiveness of phosphocholine complex and what is it to help to actually to combat fatty liver disease. Okay, so what is hepatic steatosis, or we call in general, for the layman terms, we call as fatty liver disease. It is now known as metabolic associated fatty liver disease, which the old term we normally call as non-alcoholic fatty liver disease. And there are few, and the other types of the fatty liver disease are alcoholic related liver diseases. So if left untreated of this MASLD, what will happen is that it may progress into NASH, which is the NASH which might have affiliation in liver inflammation, and followed by liver inflammation, it might cause cirrhosis, and if not treated, it may cause liver cancer. So this is a progression. So today, my topic is about how do we stop from fatty liver disease. We can stop the whole fatty liver disease so that all these chronic diseases won't occur in future. So, and it is found that actually people which are obese, or they will have a 3.5-fold of developing this MASLD, and also contributes an increase in type 2 diabetes mellitus. And besides that, we also found that the persons who have NAFLD, the fatty liver disease, will have shortened telomere length. What is this about? So in general, if you are deficient in choline and phosphatidylcholine, you will have a fat accumulation in the liver. And this fat accumulation in the liver will cause chronic diseases if left untreated. What is choline? I think you all know, you have heard of what is choline about. Choline is you normally take it from exogenous sources, and it is a precursor for acetylcholine, which is very important for your memory, for the brain function, and also phosphatidylcholine, which is very important for your cell membrane integrity. So phosphatidylcholine and choline is very important to stop the fatty liver disease. And it is found that this phosphocholine plays an important role in the upregulation of CDP pathways. This CDP pathways is very important so that you will have a balance of phosphocholine in your body. This phosphocholine is very important so that your cell membrane will stay integrity, the integrity is in there, and you have a balance, especially for your liver health. And it is helped. Today, this phosphocholine complex, I will mainly describe on 3 components, which is in my phosphocholine complex. The first one is phosphatidylcholine. The second one is alpha-GPC, and the third one is CDP choline. So these are the 3 components which is present in the phosphocholine complex. What is choline? It was formerly classified or categorized under B4, and it is very important because our own cells, we are unable to synthesize the novel choline, and we normally have to take this choline from external sources, which I will mention later what are the external dietary sources we can take choline from. And the functions are such as neurotransmitter synthesis, which you can take the precursor, choline is a precursor for acetylcholine, which is good for memory, for brain health. And choline is also important for cell membrane signaling, cell membrane signaling means that phosphocholine is the main component for your cell membrane, and it helps for cell membrane signaling for the cells to crosstalk. And it is also very important for lipid transport, especially VLDL, to maintain a balance in the body, and also for methyl group metabolism, in which it can help to reduce the homocysteine levels, so it is good for your cardiovascular health. And these are the sources for choline. You can obtain your choline from soybeans, eggs, meat, fish. These are important sources of your choline. So you must have adequate intake of these foods, then you will have enough choline. In general, choline, there are few types of choline, which is alpha GPC, PPC, CDP choline, betaine, and even phosphocholine. Today, I will present for you the pathways and the steps of CDP pathway and why it is important for the phosphocholine formation. In general, the first steps in CDP pathway is choline. So choline, you have to obtain it from external dietary sources. And this choline, when they will be phosphorylated with this choline kinase, and they will form phosphocholine. Phosphocholine will then react with the CTP, and this CTP will form CDP choline. CDP choline then will react with DAG. This DAG then will form the phosphatidylcholine, which is very important for your cell membrane integrity, for lipoprotein synthesis, such as VLDL, and also sphingomyelin formation, which is important for your nerve cells. And this phosphatidylcholine is important. That's why we need it for maintaining our liver health. And alpha GPC, this component is also a precursor for choline, needs it for the production of acetylcholine, and it also, phosphatidylcholine, can provide for the production of alpha GPC, and this can effectively cross the blood-brain barrier. So basically, our phosphocholine complex comprises of these three components, which is the CDP choline, phosphatidylcholine, and alpha GPC. And we have found numerous patents worldwide. Currently, we have obtained a few patents from different countries, which is Malaysia, Philippines, China, Australia, Hong Kong, and Macau. We have recently got the patent for this formulation of phosphocholine complex, comprising of the three components. And this phosphocholine complex, how do they work? We use the technology we call as liposomal technology. Phosphocholine complex go through liposomal technology, and they are packed within mixed micelles, and so that we ensure the micelles are transported into the liver while bypassing the reticular endothelial system and increasing the circulation time, and they will go to the target organs, they will go directly to the liver and release the contents into the liver. So you can see the mixed micelles here. Inside you have, it is loaded with alpha GPC and CDP choline, as well as PPC. PPC is the outer layer. So when it is mixed, when it is produced, it becomes a mixed micelles. And it won't easily go through your other system. It will be targeted and will go directly to your liver. And of course, we need bile acid as well. The function of bile acid is to break down the fats. And these mixed micelles consist of the hydrophilic head and hydrophobic tail. So it is like this. And let's have a look at these three components of phosphocholine. First is phosphatidylcholine, which is a vital phospholipid which is present in all of your cell membranes in your body. And its main functions is to maintain the cell integrity of your cell membranes, which helps in cell signaling. And also it is very important in lipid transport and also your lipid metabolism. And it is a precursor for acetylcholine, the neurotransmitter for your brain. And so you must make sure that you must have adequate amount of the phosphocholine in your body so that your whole CDP pathway will be upregulated. The second one is alpha GPC, which alpha GPC, just now I have already mentioned, it is a direct precursor of phosphatidylcholine synthesis, and it can cross your blood-brain barrier. So alpha GPC is good for your brain health. The last one is CDP choline. CDP choline provides choline and cytidine, and it is a choline donor and facilitates the conversion of choline into phosphatidylcholine, which is important for lipid metabolism and cell integrity. And this uptake of CDP choline will also help to regulate your CDP pathway. So in general, what is the mechanism of action of this phosphocholine complex? First, the phosphocholine complex is in the mesols, and once it is transported by parenteral route, the mesols will be disintegrated inside the liver, and the bile acid will start to activate the receptor called as FXR receptor, which I will talk more in detail in the next slide. And the phosphatidylcholine will then stimulate your VLDL, and the alpha GPC and CDP choline will be released. So everything will be in the liver itself. So what is the function of the bile acid? Bile acid has two main functions, which is to activate the FXR receptor and also activates the TGR receptor. The function of the FXR receptor is to help to repress the hepatic de novo lipogenesis, means that your liver might produce fats not only from the carbohydrates or proteins, so they will help to repress it and also stimulate the fatty acid beta oxidation. So to reduce the fat, to break down the fat contents. And besides that, they will also help to, this bile acid will also help to activate the TGR5 receptor, which helps to promote the GLP release and hence will promote the insulin release from the pancreatic beta cells, and in general will regulate your glucose metabolism. So we reduce the glucose levels. And how about phosphatidylcholine? So bile acid did their work in breaking down all the fats. Then phosphatidylcholine is then important because they are very important in lipid transport because they help to transport. You can see this is the whole phosphatidylcholine. They are very important in all your cell membranes. They are outside all your cell membranes, and they help to transport the lipids such as your triglycerides in and out of the cell within the whole cell body. Okay? And it also helps to assembly the VLDL. VLDL is very important because it helps to release and they can help to maintain your hepatic lipid homeostasis. Then after assembly of the VLDL, then it helps in the synthesis of VLDL. So meaning that the VLDL secretion, and it helps to release and budding and release of the VLDL particles from your ER and Golgi apparatus in the liver. And it helps to maintain the liver homeostasis. That's why then you won't have any fatty liver disease with phosphocholine complex. And last but not least, then the alpha GPC and the CDP choline will be released and maintain a balance in the CDP pathway. So there are actually many preclinical studies which we have conducted for this phosphocholine complex. This is an in vitro study doing the coagulation assays such as using on PT, prothrombin time, and the fibrinogen, because as you know, if patients with non-alcoholic fatty liver disease or type 2 diabetes mellitus has some coagulation problems. So this is a project done by CRC Sabah, and it is already completed. And we use 20 healthy patients, and group 2 is with normal BMI, not diagnosed with fatty liver, and group 3 is with type 2 diabetes mellitus and with overweight, BMI, and NFLD, and we use paired t-tests. So what was the outcome is that the administering of this phosphocholine complex led to a notable improvement in all PT outcomes. It actually helps to normalize your prothrombin time. So it means that this phosphocholine complex can help to aid in the patients with these coagulation problems. And it also helps to decrease the fibrinogen level significantly in all groups. Because as you know that type 2 diabetes mellitus patients will have some problems in their fibrinogen levels, which they have high levels. And besides that, we also did an in vitro MTT assay, which this is on the cell culture. We did it on the liver cells. And we found that there's a concentration of 0.5% of this phosphocholine complex does not cause any cytotoxic potential on the cells, so that it is deemed safe to be used. And we also checked for the triglyceride assay to see whether this phosphocholine complex can actually reduce the triglyceride content. And 0.5%, it is found that you can see no treatment, the millimole is 1. And with 0.5% of this phosphocholine complex, we have already reduced the TG content. So that this phosphocholine complex is actually effective in removing the triglycerides. We also did OIRAT ore staining. OIRAT ore staining is something like you induce fat into the liver cells, and then you treat it with the phosphocholine complex and see whether is the fat removed. So what did we do is we use the liver cells and we expose with the fat, which is the oleate and palmitate, with some ratios. Then we wash it with PBS, and then we add a different concentration of the phosphocholine complex. Then we will stain with the stain, we call it as OIRAT ore, so we always call it as oral staining. Then we examine within the fluorescent microscope, and this is what we found. This is the normal HepG2 cells, the normal cells, liver cells, and we induce steatosis in the HepG2 cells. You can see the red color means that you have oil inside, and we use different concentration, and you can see the oil is reduced in 0.5. So that we can see that phosphocholine complex can enhance the treatment of NAFLD by decreasing the buildup of fatty substances in the liver. And we also did a network pharmacology analysis, so means that to elucidate what type of pathway is actually involved with these 3 components. And we use the database, we use the Swiss target prediction database, and we assess what type of biological pathways are involved. And we use the gene ontology and KUGG pathway enrichment analysis using string version. And we found that the biological pathways involved in phosphocholine complex are PPAR and AMPK signaling pathways, which these 2 pathways are predicted for reducing the fibrinogen level and also reduce the oil content in the liver. And this activation of these 2 pathways can improve insulin sensitivity and also reduce inflammation, promote adipocyte differentiation, and leading to a reduction of hepatic fat accumulation. Okay? So, and this AMPK is also shown to improve the liver function and reduce the progression of NAFLD in animal models. Besides that, on in vitro studies, we also started to do some animal studies. These are called the dose range finding studies, and also the maximum tolerated dose as a safety studies for the SD rate rats, and the study is completed. And we did a few groups of phosphocholine complex. First one is the control group. The second one is using 1.25% of phosphocholine complex. Second one is 2.5%, and the fourth group is 5%. And the rats were administered IV, and then we waited for 24 hours, and then we did an autopsy and checked, is there any lung congestion, is there any liver problems within the rats? And we found that there is no mortality and no significant adverse effects for the rats itself. All these studies are conducted within an IBM, which is a certified GLP lab under OECD guidelines. And of course, since we have already found the percent, all of these concentrations are good. That's why we decided to go for a maximum tolerated dose. We wanted to see what percent can the rats go to. Then we tried another 3 more groups, control group, and the phosphocholine complex. We used 5% to 10% to 20%. And we did an IV administration as well. Then we waited for 14 days to see is there any problems with the rats. And we found that the MTD is below 20%, meaning that the best concentration for now for phosphocholine complex is 10% for the rats. So now the further studies we will continue for acute and subchronic studies for the phosphocholine complex. So in conclusion, phosphocholine complex is very effective in managing fatty liver by enhancing the lipid clearance and inhibit your fatty liver production, and also with anti-inflammatory benefits, and also helping the repair of the hepatocytes. And this phosphocholine complex can be served as a holistic approach to address the root cause of fatty liver disease, promoting your liver health, and helping to further continue into progression into chronic disease. So this phosphocholine complex comprises of PPC, alpha GPC, and CDP choline. Thank you.